All right, thanks everybody for joining us today and for sticking around for the last day of the conference. My name is Stephanie Young. I'm currently a Director of Bioinformatics at Roche Sequencing Solutions. Today I'll be sharing with you data from an evaluation of the AVENIO Tumor Tissue Comprehensive Genomic Profiling, or CGP kit. This is a disclaimer slide just to say that the content and opinions presented today are my own professional views and don't necessarily reflect those of my employer, Roche. And also please note that the AVENIO Tumor Tissue CGP kit is for research use only and not for use in diagnostic procedures. In case I run out of time, I did want to acknowledge upfront my colleagues listed on this slide who conducted the evaluation study and generated the results. Today, I'm really just presenting the data today on their behalf. Today, I'll be covering 3 topics. First, an introduction to comprehensive genomic profiling. Second, an overview of the study and methodology. And third, the agreement results between the AVENIO Tumor Tissue CGP kit and the Foundation 1 CDX assay. To put my talk in context, I wanted to briefly review the concept of precision medicine. As many of you know, precision medicine can improve patient outcomes by identifying the right drug at the right time for the right patient. Cancer treatments have really evolved over time from a one-size-fits-all approach to now more personalized treatments tailored according to patient-specific information, including biomarkers. In a conventional approach, shown here as a hypothetical example on the slide, let's say a therapy is given to a general patient population and we observe an overall response rate of 30%. In a precision oncology approach, research studies are done to see if of the patients who did respond, did their tumors share any common molecular characteristics, such as a specific mutation in a cancer gene. This type of information can lead to biomarker discoveries and new targeted therapies. And when a targeted therapy is given to patients whose tumors have that corresponding biomarker, you would expect higher overall response rates, let's say 75%. And patients who don't respond to that treatment might make up other molecular subsets who could then benefit from other targeted therapies. There are many examples of these molecularly targeted therapies. I'm sure this audience is familiar with these, but one well-known example in lung cancer is targeting specific mutations in EGFR. Patients with advanced non-small cell lung cancer, if they have these EGFR activating mutations in their tumors, they benefit from receiving EGFR inhibitors, such as erlotinib. Clinical trials have shown that there's a 60 to 80% reduction in risk of progression or death if patients receive erlotinib compared to standard of care chemotherapy. Besides EGFR, there are many targetable biomarkers. In metastatic non-small cell lung cancer, there's been a large increase in the number of available targeted therapies. And if you just look on the slide, back in 2015, only 2 genes were listed in the NCCN guidelines as having available targeted therapies. Today, there are many more genes, including ROS1, BRAF, and KRAS. So given this large expansion in molecular biomarkers in oncology, the ability to test for multiple genes at once is needed and enabled by next-generation sequencing. Not only can different genes be tested for at once, but different types of mutations. And this is important because just of the genes listed on this slide, the mutations of interest cover a variety of variant types, including point mutations, insertions, deletions, and rearrangements. And so comprehensive genomic profiling, powered by next-generation sequencing, helps researchers gain insights across different genes and variant types, and this can further advance precision oncology. Now I'd like to introduce the AVENIO Tumor Tissue CGP kit. This is a distributable, research-use-only kit for tissue samples. The CGP kit leverages the AVENIO workflow, which includes optimized DNA extraction, library preparation, and targeted enrichment. The hybrid capture panel in the CGP kit is the same content as F1 CDX. The CGP kit also leverages the Foundation-1 secondary analysis platform, which is cloud-based and accessed through something called the AVENIO Connect software. The bioinformatics pipeline has been optimized to work with this lab workflow, and it also uses a Foundation Core, which is a database of real-world CGP results from over 500,000 clinical samples. Overall, from DNA extraction to variant calls, this is a 5-day workflow. The analysis pipeline is able to produce variant calls as well as genomic signatures. Variants include single nucleotide variants, multi-nucleotide variants, insertions, deletions, rearrangements, and copy number alterations. The analysis also provides scores or values for 3 genomic signatures, specifically tumor mutational burden, microsatellite instability, and genomic loss of heterozygosity. This slide just shows that all 324 genes in the Avanio tumor tissue CGP kit panel matches the same content as the Foundation-1 CDX panel. So with the CGP kit, the hope is that every lab can now have the choice to perform comprehensive genomic profiling with the same analysis power as Roche FMI. And hopefully this can help further advance cancer research discoveries. So now onto the setup of the evaluation study. This was a scientific study to assess the alignment of detected genomic variants between the 2 different assays, the Avanio tumor tissue CGP kit and the Foundation-1 CDX assay. FFP tissue samples from solid tumors first underwent DNA extraction, and then aliquots of the same DNA extraction underwent the CGP kit assay, which was performed at Signature Diagnostics in Potsdam, Germany, as well as the F1 CDX assay performed at Foundation Medicine in Penzberg, Germany. The data I'm presenting today is an initial comparison of the first 50 samples processed so far. These have been mostly from breast, colon, and prostate cancer, with a few from melanoma, head and neck, and then bladder, ovarian, and stomach cancer. The first thing we looked at was, even though the same amount of sequencing was performed, actually there was higher coverage obtained using the CGP kit. So you'll see on the slide that the mean coverage of the F1 CDX assay was 460x and ranged from 260 to 940x. For the CGP kit, the mean coverage was 4 times higher at 1,870x and ranged from 1,080 to 2,400x. The difference in coverage is due to the different assay chemistries and bioinformatics pipelines, which I'll touch upon a little bit later. Next, I did want to take some time to describe how we generated the agreement metrics when we compared variant calls between the 2 different methods. So to start off, shown here is a 2x2 contingency table. If a variant was called in at least 1 sample in the study, it was included in the calculation. And then across all samples, all included variants were tallied here as either called or not called by each assay. The first metric we calculated was something called overall percent agreement. This is taking the sum of agreed positive calls, which would be A in the table, and then the agreed negative calls, which would be D in the table, and then dividing by the total number of calls. This metric accounts for both positive and negative agreement and is an unbiased way to measure agreement since we weren't taking either method as the truth. This is simply a way for us to see how well aligned the variant calls are between the 2 different assays. Next, we actually did look at average positive agreement, and this was calculated by looking at the number of agreed positive calls, again, A in the table, and then dividing by all positive calls made by either method. So this would be A plus B plus C. Again, we're not taking either method as the truth, so this is a little bit different calculation than what folks are used to seeing, such as PPA or NPA. But we did believe that this was an unbiased way to look at alignment between 2 different methods in terms of positive calls. And similarly, we calculated average negative agreement. So this is the number of agreed negative calls, D in the table, divided by all negative calls made by either method, which would be D plus B plus C. So this is, again, to measure the alignment between the assays in terms of negative calls. Okay, and if you'll bear with me with one more mathy slide. So for genomic variants, we took a slightly different approach. Although the AVENIO tumor tissue CGP kit does report numeric values for TMB, MSI, and genomic LOH, we actually applied thresholds to generate categories for these 3 genomic signatures for the evaluation. And so these categorical results might have more than 2 levels. In other words, instead of a variant being called or not called, something like TMB would have 3 levels, a TMB low, intermediate, or high. And so this type of analysis requires a larger contingency table, and shown here is an example of a 3x3. And these types of larger tables can be evaluated using something called inter-rater agreement metrics. And one well-known metric is Cohen's kappa. So Cohen's kappa corrects for how often 2 raters might simply agree by chance. And we also took a look at the weighted version of the Cohen's kappa, otherwise known as weighted kappa. So this further accounts for the degree of agreement or disagreement between ranked categories. So this just means that if one method calls TMB high and another method calls TMB low, that's a greater disagreement than if one method called TMB high and another method called TMB intermediate. Okay, so using the metrics I've described so far, let's take a look at the agreement results between the AVENIO tumor tissue CGP kit and the F1CDX assay. So first, we actually distinguished between complex variants and short variants. Short variants are those restricted to 1 exon, and so these would include SNVs, MNVs, insertions, and deletions within an exon. Complex variants cross exon borders, and so these would include deletions spanning more than an exon, as well as truncations, fusions, or rearrangements. The number of short variants detected in each sample was very similar between F1CDX and the CGP kit. The Pearson correlation was 0.95, and both assays had a median of 9.5 and a mean of 18 to 19 short variants detected per sample. Compared to short variants, fewer complex variants were detected per sample. Here's a table comparing the counts between the 2 assays. Most samples actually did not have any complex variants detected. And the most frequently detected complex variant was TMPRSS2-ERG, which is a fusion found in about 50% of prostate cancers. And this is not surprising given that in the first 50 samples analyzed so far, 12 of them were from prostate cancers. So next, we calculated the agreement in calling genomic variants between F1CDX and the CGP kit. We found that there was a high overall percent agreement between the 2 assays in terms of calling short variants, complex variants, and copy number alterations. This agreement was 98% or higher. And if we focused only on the positive agreement, we found that it was 92% for short variants, 79% for complex variants, and 65% for copy number alterations. And we believe the reason for the lower percent positive agreement for complex variants is the limited number of complex variants that were detected, as shown on the previous slide. Only 18 out of the 50 samples had any complex variants detected. Also, the higher coverage of the CGP kit generated more complex variant calls than the F1CDX assay. As for the copy number alterations, the bioinformatics pipeline is different for that class of variants and could have also contributed to the lower positive agreement between the 2 assays. Okay, and then if we focused only on the negative agreement, we found that it was 99% or higher for short variants, complex variants, and copy number alterations. Now on to the genomic signatures. So the first one I'll cover is genomic LOH. The CGP kit provides both a score and a category, microsatellite instability. So the CGP kit provides both a score and a category for MSI. The score is the fraction of unstable loci out of all of the evaluable microsatellite loci. And so based on the score, a classification is made for each sample in terms of MSI high versus microsatellite stable. You'll see on the slide that between the two assays, the MSI status was identical for all 45 samples for which there was an MSI status evaluable by F1-CDX. And of these 45 samples, 2 were MSI high and 43 were MSS. Okay, so this is the genomic LOH. So for this one, the CGP kit reports a score for genomic loss of heterozygosity. This score represents the fraction of LOH in the tumor genome, and it's based on the genome-wide copy number profile as well as the minor allele frequencies of germline SNPs. The same cutoff was used for both methods to classify genomic LOH low versus high, and a failed status was reported if the genomic LOH could not be reliably determined, such as due to an unusual copy number profile. We found that between the two assays, the genomic LOH status had a weighted kappa of 0.82, which can be considered strong agreement. And finally, the third genomic signature we evaluated was TMB. So this is reported as mutations per megabase and counts the number of somatic, passenger, non-synonymous, and synonymous variants above an AF of 5%. So for this analysis, samples with up to 5 mutations per megabase were considered TMB low. Those with between 6 to 19 were considered TMB intermediate. And then for those with 20 or more mutations, those were considered TMB high. An unknown status was reported for samples where coverage may have been low, or there was either very high or very low tumor purity, which makes the TMB calculation less reliable. We found that between the two assays, the TMB status had a weighted kappa of 0.88, which is also considered strong agreement. In summary, we did find a high degree of alignment between the AVENIO tumor tissue CGP kit and F1-CDX for all of the genomic signatures and variant types. Specifically, I wanted to highlight the overall percent agreement, which incorporates both positive and negative agreement. We observed 98% or higher overall percent agreement between the two assays for short variants, complex variants, and copy number alterations. The weighted kappa coefficient was 0.88 for TMB and 0.82 for genomic LOH, and these indicate strong agreement. And the MSI status was identical between the two assays. Notably, on the same 50 samples, higher coverage was obtained using the CGP kit compared to F1-CDX. And this is a result of differences in the chemistry and the bioinformatics pipeline. And I know I keep mentioning differences in assay chemistry. So on this last slide, I did want to point out a few of those key differences. In the CGP workflow, there were enhancements made to better isolate, repair, and QC the DNA derived from FFP tissue samples. So first, the DNA isolation was optimized to yield higher quality and quantity of DNA compared to typical column-based techniques. Second, a DNA damage repair step was introduced to minimize cytosine deamination errors, which is a major cause of noise in NGS data derived from FFP tissue samples. When oxidized, the cytosine becomes a uracil, and that's actually read out as a thymine. So in the sequencing data, this will show up as C to T or G to A errors. The inclusion of a DNA damage repair step helps address this by cleaving the uracil from the DNA and thereby reducing the overall observed C to T or G to A error. And finally, the third workflow enhancement is a DNA quality check to help ensure that there's sufficient amplifiable DNA going into library prep. This QC step is based on a qPCR that compares a long amplicon to a short amplicon. Damaged DNA will tend to have shorter fragments, so the ratio of long to short amplicons is smaller. And then this ratio is actually used to calculate a minimum input mass requirement to help ensure successful library prep. All right, thanks for your attention. I'd be happy to take any questions now.
Thanks, Stephanie. We do have about 3 minutes for questions. Jane?
Yes, very quick one. Let's see, for your positive your positive comparative rate was around 91%, if I remember correctly, for the short SNVs and indels. So my question is, of those that were discordant, the 9%, were those predominantly SNVs or were they indels?
Yeah, so actually, I don't have that information, but I can look that up and then follow up with you offline.
Thank you.
Thank you.
Stephanie, there is an online question. To reach 95% confidence per literature, Jennings et al. Assay requires 59 samples. Is there a reason lower samples were included in the Avenio validation?
Oh, okay. I see. Yeah, so sorry, this is not a validation. It was simply just an initial look at the first 50 samples. The team is actually hoping to generate over 100 samples in the final study. This is just the sort of the first snapshot. Yeah, so it's not a validation, not the 59 samples that people tend to use for that. So I hope that clarifies the question. Yeah.
Okay, thank you. We're going to thank Stephanie, and we're going to ask, I think, Tui to come up as we prepare for the next session. Thank you very much.